The overall goal of this procedure is to isolate 3D in vitro basement matrix-embedded intestinal organoids and to subsequently fix and immuno-label the microtubules and centrosomal proteins.
3D in vitro organoids are ideal models for studying key biological questions in cell and developmental biology. But localizing endogenous proteins and structures in 3D models is more complex than in 2D cultures. Importantly, the fixative needs to preserve the delicate 3D architecture while also preserving the antibody antigenicity. The presented methods include isolation, fixation, and immuno-labeling of 3D in vitro intestinal organoids, but the fixation and immuno-labeling protocols can also be used in ex vivo isolated tissue. The main advantage of the presented formaldehyde methanol fixation protocol is that it preserves the 3D structures while also preserving antigenicity. It enables good penetration and clearance of both fixative and antibodies for effective labeling of microtubules, actin filaments, end-binding proteins, and several centrosomal proteins, including ninin. Demonstrating the procedure will be Dr. Deborah Goldspink, a former postdoc in my lab, and Dr. Zoe Matthews, a co-author.
Following the isolation of intestinal crypts and villi, isolated epithelial structures can be fixed for immunostaining. The isolated crypts can alternately be seeded into matrix domes, which will then form